Hello, my name is Mengya Zhang. I'm from Dr. Qingyu's lab. We're at the Institute of Neuroscience, Chinese Academy of Sciences. Today I'll be talking to you about a combined EEG and fMRI study we did that looked at the neurogeometries of abstract working memories. Working memory has been an intensively researched area for past decades. The main focus, at least in the cognitive neuroscience perspective, has been on specific content, and within visual research that would be angles, colors, objects, sometimes even words. However, the neuromechanisms for maintaining abstract higher order task informations are relatively unknown. And by abstract information, I mean information like task goals, rules, or contexts, things that govern the usage of low-level sensory information. What we do know is that in contrast to specific contents, representing abstract working memory often involves more higher-level association regions. And understanding how the brain represents task-related abstract information and the interactions with specific contents in a goal-directed fashion, while both being held in mind, is the key to understand complex and flexible cognitive behaviors. And the challenges to researchers were twofold. So firstly, neurons in these higher level cortices do not show simple tuning selectivity the same way neurons in early visual cortex show. For example, the orientation tuned neurons in V1 or the color tuned neurons in V4, among other areas. And secondly, when people do study abstract task information and their representations, they often use qualitatively different conditions that make it difficult to test the neural coding principles adopted by these representations. With those questions and challenges in mind, we designed a novel delayed continuous recall task in which the participants first needs to remember a task goal, followed by a sample stimulus, and at the later response period, they need to recreate the stimulus color and size according to the goal. And most critical to our design is that both of the task goals and the stimulus, the sample stimulus features formed a two-dimensional task space, which can act as a template to compare the representational geometries with. So for the stimulus, the color and size are sampled from a continuous range of values. So it can be from small to big and from very green to very red. And the goal space goes along with the stimulus space in that it can be any combinations of changing the size and the color to either directions. So the task goals themselves form a two-dimensional space as well. And to give you a pair of concrete examples, the objects at the center are the starting stimuli for the participant to memorize, and depends on the goal of the current trial, the end products the participants need to recreate are at the four corners. Very importantly, the distances between the starting and the end stimuli are fixed and pre-learned in a behavioral session, so that for each trial we have a correct answer and we can derive memory error from it. And here's a demo of how the trials actually run. At the beginning, the participants see a cue that represents the goal of the current trial, followed by a delay, and then they see a sample stimulus that they need to remember the color and size of, and followed by another delay, and then the response period begins when they need to adjust the stimulus feature to match what they think is the right answer. To have a way to compare the geometry in this high-dimensional neural state space with the hypothesized task space, we first used a supervised PCA to find the first two principal axes and projected the data onto the subspace. And then we used a measure called circularity index to quantify the similarity between the neural and the theoretical geometries. And circularity index has a very simple equation. It is 4 pi times the ratio between the area of the shape and the square of the perimeter. For a perfect circle, the circularity would be 1, and evidently it is affected by the number of points or edges in the shape. For a structure with only 4 points, such as the task space, the task goal space, the maximum value is achieved when it's a square and it's around 0.8. And with this metric, we can perform significant testing with permutation procedures, and circularity index would be the metric we use throughout our analysis as a measure for our two-dimensional geometry. We divided our analysis between the frontal and posterior EEG channels, and here it first shows the group level Go geometry time course in frontal channels, and the red line represents the significant threshold. It seems that only during delay 1 it has an extended period of time when circularity exceeded the threshold. And if we take the average delay 1 data out and project it onto the low-dimensional subspace, it looks like this. And hopefully you can see how much it resembles the designed task goal space. For stimulus representations, the only period of time that exceeds extensively the significant threshold is during sample presentation in posterior channel activities. And you might notice the threshold here is different from the Go geometry. It's because we used 6 points to calculate the PCA, and the significant threshold is affected by the number of points. So what we found is that for the 2D Go structure, the only significant time is during the first delay in frontal channel activities, and for stimulus, the only significant time is during sample presentation and only in posterior channels. Next, we wondered whether the similarity of these 2D neural geometries with the hypothesis ones would predict memory performance. So we used a representational similarity-based multiple regression approach to assess the relationship between these neural geometries and the trial-wise variations in memory error. So for Go geometries, which is on the left, behavioral relevance arose first at frontal channels, denoted by the blue line, and cascaded to posterior site at the end of the first delay. And for sample stimulus, only the 2D structure within the posterior activities predicted behavior. So what could be mediating the frontal to posterior transition of the 2D Go geometry? For that, we investigated the frontal medial to posterior theta coherence, or FMT, which has been proposed to play a critical role in sending top-down control signals during memory delay. And for reassurance, in our data, we indeed found significant theta coherence clusters. So the FMT cluster at delay 1 is significantly correlated with the posterior Go circularity at delay 2. And the FMT at delay 2 predicted the sample structure at the same time. And you might be thinking, why is this second relationship negative? We think it's because the sample information needed to be transformed into a response representation in preparation for the response period. So we did a correlation with the response representation, and here it actually shows a positive relationship with that. So the relationship between the sample and the response is kind of like an ebb and flow kind of dynamic. And to make this part of the result a bit clearer, what we think happened is that the goal representations was transmitted through FMT from frontal channels to posterior channels where the sample representation is stored. And the response representations also formed frontally and relied on the FMT to modulate the sample stimulus representation. So to harvest the superior spatial resolution of fMRI, we had participants perform the same task in scanner and repeated the PSA-based circularity analysis in predefined ROIs. And these ROIs cover the entire prefrontal cortex and visual cortex as well. So long story short, the result is quite consistent with EEG findings. We found significant 2D goal geometry in dorsolateral PFC and medial PFC in delay 1. And in delay 2, it is only significant in one ROI of the visual cortex, and that's in the dorsal visual stream, which roughly translates to V3A, V3B, and the region immediately superior to that. And similar for the 2D sample geometry, we found it is only significant in primary visual, or V1, during delay 2. And to sum up our results, abstract goal-related working memories are represented in a low-dimensional subspace that preserves the designed task space. And this representation is first in prefrontal cortex when it's first encoded, and when it needs to be applied with concrete information, it is transferred to visual cortex. And both 2D goal and stimulus geometries are important to memory performance. And lastly, frontal medial to posterior theta coherence, or FMT for short, serves as a potential mechanism underlying the interaction between abstract and specific working memory. And I want to thank my founding parties and my supervisor Qingyu and everybody in the Visual Cognition and Memory Lab at the Institute of Neuroscience. And thank you very much for listening.